to my Paleoclimate proxy paper. I will be talking about chemical proxies, and my name is Peter QU. Paleoclimate proxy is a natural recorder that can be found in ice cores, tree rings, or sediment layers that preserves information about past climate conditions, allowing scientists to reconstruct past climate patterns. Today, I will be talking about chemical paleoclimate proxies that are geochemical signatures that are often measurements of chemical elements preserved in natural archives that could be analyzed to reconstruct past climate conditions. Proxy, I will be talking about four chemical proxies, which are stable isotope ratios, trace element ratios, organic biomarkers, and ice core gas analysis. Stable isotope ratios are the ratios of stable isotope of the same element with different neutron counts that are not radioactive. Stable oxygen isotope ratios, oxygen 18 was used in European alpine trees to reconstruct howling summer hydroclimate, revealing long-term aerification trends and demonstrating the utility of isotope proxies in paleoclimate research. The biggest strength of stable isotope is that it is stable and it does not decay, and they are naturally occurring in all environments and organisms. The biggest weakness of this proxy is that the proxies require advanced instruments and difficulty in quantification that requires specialized expertise. Trace element ratios are obtained from geological archives widely used to infer paleoclimate information. For example, strontium to calcium ratio in coral skeletons correlate with sea surface temperature due to its relationship with temperature during the carbonate precipitation process. This proxy is critical for deciphering geological processes in the upper mantle and the genus of the continental crust, but its critical weakness is in difficult tectonic settings. It could be difficult to source identification, necessitating the need of integration or with additional isotopic data for accurate interpretation. Organic biomarkers are found in marine and lake sediments, ice, and spiliforms. This proxy could give temperature, precipitation, and vegetation changes about the past environments. An application of this proxy is the alkanone, lipid produced by the hepafine algae. Based on alkanone's unsaturation index, the sea surface temperature can be precisely determined. Organic biomarkers can provide insight into carbon cycling, primary productivity, and sedimentary processes. What limit this proxy is the challenge of preservation. Biomarkers with low preservation capacities like the lipids may degrade rapidly in samples, resulting in unreliability for long-term paleoclimate reconstructions. For gas analysis can provide direct measurements of past atmospheric composition, and from that we could make direct inferences about other past climate variables. Ice cores can be drilled from polar ice sheets and glaciers. The air bubbles trapped inside the ice lattices, contain samples of ancient atmosphere. The gas concentration collected are compared to ice age models to build timescale for all data. The biggest limitation of ice core analysis are gas mobilization in the ice can blur high frequency signals, reducing resolution. And ice didn't occur in all periods of the Earth, so this proxy is limited to the periods that had ice. Last, we will talk about the case study using astrocod shell chemistry as a proxy. Astrocod are small crustaceans inhabiting in oceans. Construct their shells through biomineralization, obtaining and storing elements from surrounding water. Astrocod actively regulate the uptake of ions from water to form their calcium carbonate shell. This regulation is influenced by environmental factors like temperature, salinity, pH, and the availability of trace elements. Normally, we will use oxygen and carbon isotope ratios to study them, but recent advancements in analytical techniques allow us to study trace element ratios inside Ashika shells. The modern analytical tools like laser ablation, inductive coupled plasma mass spectrometry, and nanosecondary ion mass spectrometry enable high-resolution analysis into Ashika shells. Trace elements like cadmium and zinc can provide insight into past productivity and nutrient conditions. This is still an ongoing research focus on refining the calibration of trace elements and investigating further species-specific variants of element uptake and exploring their applications across diverse geo uh, settings. Thank you for watching my presentations. These are my sources.